So we're going to start with a traditional summary of the science part of AASLD. We have 4 speakers, and Mark, Sarah, James, and Sammy. They will all of them discuss what they think was most remarkable in terms of different studies that were presented within AASLD, and feel free to ask questions or comments.
Yeah, we kept some room for discussion, but I mean it can be done just in between. I selected three studies. I'm doing the more clinical and translational part, whereas my colleagues are gonna go more basic. So the first one is entitled Combined Methylated DNA and Protein Markers, an accurate blood-based test for early-stage detection of HCC, which was presented by Naga Chalasani from Indiana University, who actually was here in Mount Sinai earlier this year. Well, as you all know, among the cirrhotic population in the United States, there's only a minority of them, around a third, that are aware of having liver disease, and actually just a minority of them, between 5 and 10% of the total, are enrolled in surveillance. And on the other hand, we know that even among those patients that are enrolled in surveillance, only a minority, around even less than half of them, are detected at early stage. And this is because the techniques we use nowadays, which are ultrasonography or alpha-fetoprotein, have a very low sensitivity. So considering that, there's a clear need for new surveillance approaches. So the authors of this study want to identify a panel of methylated DNA markers and different proteins, including AFP, to detect early-stage HCC. So what they do is a case control study. They collect blood samples from 28 different sites with patients with HCC and match controls according to age, patients with benign liver disease. And what they check first is this panel of 10 methylated DNA markers. This is a panel that they come from a publication from the same group earlier this year in hepatology. So they use these markers that previously have been related to HCC and combine them with different proteins, as I said, with mostly alpha-fetoprotein. And then what they do is a logistic regression analysis to make either positive or negative HCC calls. So going directly to the results, they have a total of 137 HCC cases, majority male and with cirrhosis as expected, and more than 300 controls. As you can see here in this plot, around half of the cases are early stage, VCLC 0 or A, whereas all the remaining ones are from B to D. And these are the results. What they show is that a combination of 4 of these methylated DNA markers plus 2 protein markers, which are AFP and lectin-bound AFP, are the combination of the 6 is the one yielding the higher sensitivity and also specificity for HCC detection. As you can see, pretty nice. For early stage, 70%, and for all stage, 80%. I didn't mention that, but they pre-specify the specificity at 90%, which means that they discard all the options with less than 90% specificity. And as you can see here, they compare this combination with AFP at different thresholds. And I wouldn't say not surprisingly, the sensitivity of this combination is higher and the specificity is maintained, as I said, more than 90% for the combination, with a very nice area under the curve of 0.91.
So this is the Exact Sciences platform?
I'm not sure, yes.
And the ones that make Cologuard.
And so moving to the conclusions, reading textually from what the authors say, a panel of 4 methylated DNA markers and 2 proteins shows high sensitivity for early-stage HCC, and these data support the near-term potential for improved performance of this blood-based testing for HCC detection. As a bit of a discussion, I would say that I'm missing here the comparison to the standard of care, which is not AFP alone, but AFP plus ultrasonography. So of course they show that this combination of markers is better than AFP alone, but I really would like to see how it compares to the standard of care. Moving to presentation number.
Before you go on.
Yes.
Augusto, you're the expert. Is there anything technical that makes this a very straightforward technology?
If you go back, so there are 2 big companies running studies on DNA methylation in the blood. This is one, and so the technology is straightforward. The performance is decent. I wouldn't say it's outstanding, but it's decent. For example, for early stage, sensitivity is 70%, which is good, but it's not outstanding compared to the 63 that is reported for AFP. And ultrasound. Another thing that's interesting that I asked Naga in the talk is, so the sensitivities when they combine the methylation markers and AFP, but how much are the methylation markers increasing the performance of AFP? Is there a significant increase on that? And he said yes, but it's not, quantified precisely. What are we adding with the 4 methylation markers? But definitely there's huge interest in developing methylation.
What about etiology, like etiology specific? Anything like etiology based?
No.
Risk of
I mean, to go faster, I said to go faster, I didn't include the etiology information. They show that there are no differences according to etiology here.
In terms of terminology, would this be considered liquid biopsy? Or is that
Maybe I was going to have better,
Yeah,
but I would say yes, it's from the blood. So.
Yes. We try to capture methylated reads that are released by the
Mark, did they divide their cohort to see, to do some test samples and some training?
I mean, training and validation? I don't think so. At least it's not clear.
I'm
Right?
afraid in that case that could be overfitting.
Absolutely. Okay, so moving to study number 2. It's entitled Immune Changes During the Early Steps of Human Liver Carcinogenesis. This was presented by the group of Julian Calderaro, which is a member or former member, I'm not pretty sure, of the Jessica Sumpen-Rossi lab in the INSERM in Paris. So as you all know, HCC is the prototype of an inflammation-driven cancer. As in the background of liver cirrhosis and also in the background of chronic liver disease, there is inflammation, and this inflammation is it's what, together with other factors, triggers the development of HCC. So this is pretty well known. And on the other hand, we know that the accumulation of mutations in these early steps of liver carcinogenesis, from the cirrhotic liver to dysplastic nodules and later HCC, is a pretty well-defined process. That it's an accumulation process. But however, the immune modulations that occur in these early steps of liver carcinogenesis is completely unknown. It has never been characterized. So this is the aim of the authors. And to do that, they collect more than 125 macronodules. Here, my first concern, it's not very clear how they define macronodules. They don't define a size. I would assume that these are more than 1 centimeter, but I'm not totally sure. So, and this is the breakdown of these nodules. They have 31 with low-grade dysplasia, 18 with high-grade dysplasia, then 24 which are already early HCC, and 55, almost half of this group, which actually are small unprogressed HCC. So these are not really unspecific nodules, because you have like more than half of them being HCC, and I assume that these nodules have to be more than 1 centimeter. Then what they do to characterize these immune changes is they do a multiplex immunohistochemistry to assess the density of different immune cell populations. And then they do it in a subset of lesions of around 30. They do a panel of gene expression profiling using nanostring. And then what they do is to normalize these values according to both immunohistochemistry and gene expression. They normalize according to the adjacent non-specific nodules, meaning that, to my understanding, to the cirrhotic tissue. So going to the results directly, they show an increased density of T cells, B cells, and dendritic cells in all stages, meaning that this immune infiltration appears in early stage of this HCC development, even in low-grade dysplasia nodules. And they show, on the other hand, that these TLS or tertiary lymphoid structures are identified only in these lesions with high-grade dysplasia and actually being HCC, but not in low-grade dysplasia or in non-specific nodules. Then, using the gene expression profiling, they identify a chemokine signature using these 12 chemokines. And as you can see here, the patients on the left-hand side harboring this signature independently of the grade. You can see here the grade is not very well. The quality is not very nice, but you can see that the signature it's present and independently of the grade of the nodule. And they show that the patients in this subset harboring this signature show as well upregulation of of all these pathways, which are antigen and presentation, T-cell markers, B-cell marker, and so on. And interestingly, as well, exhaustion markers and the expression of immune checkpoint molecules such as TIM3, PD-L2, and CTLA4. So with all that, they go to the conclusion that anti-tumor in situ immune responses develop during the very early steps of liver carcinogenesis, and they identify this as a small subgroup of lesions showing the expression of immune checkpoint molecules, which could favor immune evasion and allow the progression to a fully developed HCC, with the implications that this might have for anti-HCC chemopreventive strategies. As a discussion here, feel free to add whatever you think, but for me, the first point was it's not clear how they select the macronodules according to the size or according to incidental findings. I don't know. Then I would like to know if there is increased inflammation in the macronodules, but not in the surrounding cirrhotic nodules, because they show that this is only in the nodules because they normalize according to the surrounding tissue. So which is the rationale for chemoprevention when these nodules already appeared? So I think that chemoprevention strategy should be based on the findings in the cirrhotic tissue, not in the nodules. And last, I would like to see as well if the different macronodules in the same patient show the same expression profile, because again, the chemoprevention should be based on patients and not on nodules. And very fast, just a last study, clinical one, the Checkmate 040, comparing the performance of Nivolumab and ipilimumab combined in patients with advanced HCC after sorafenib treatment. You all know about the promising results of Nivolumab in the phase 2 trial to treat advanced HCC. Unfortunately, this was not confirmed in the phase 3 trial. And here it comes the rationale for the combination of these two immune checkpoint inhibitors, because it's, for example, in other cancer types such as melanoma, it's the standard of care. So they designed here, the authors, a phase 1b/2 study to treat patients with advanced HCC in second line after sorafenib treatment. These are the 3 arms of the study. As you can see, the combination is of both drugs, but in different doses. The first arm is Nivo at low dose plus Ipi at high dose. Arm B is the other way around, Nivo high dose plus Ipi low dose. And arm C is different strategy according to the follow-up of the patients. So what they do is they continue the treatment as usual until toxicity or disease progression. And the primary endpoints are safety and tolerability, where overall survival and overall response rate are secondary endpoints. They randomized a total of 148 patients with pretty much advanced liver disease, 82% extrahepatic spread and 91% BCLCC. And these are the results. I would say nice for arm A with a median overall survival of 23 months. And for the other two, around 12.5 months. So here they show that overall response rate are a little bit higher. They say double, I wouldn't say double, but they are higher than the ones reported for Nivolumab alone, that that are around 20%, 18 to 20%. So these are around 25 to 30%. And they show as well the rate of immune-mediated hepatic adverse events. They show that around 10 to 10% in the different arms of patients developed these immune-mediated adverse events, but the vast majority of them resolved pretty fast in the next 6 weeks approximately. All of them were treated with high-dose prednisone. And what is more interesting to me is that patients that presented these adverse events and that were rechallenged with Nivo or Ipi did not present recurrence of the adverse event. So the conclusion here is that the combination of Nivolumab plus ipilimumab showed durable responses and a manageable safety profile in patients with advanced HCC treated with sorafenib. So this is, of course, the first study reporting combination of these drugs in advanced HCC. We'll have to see the results after this expected change of paradigm in first line, after we know that the atezolizumab plus bevacizumab has positive results. Regarding this study, I'm missing, I know it's a phase 1b/2, but it would have been nice to see a control arm with Nivolumab or so that with the drug in monotherapy. And as well, the authors don't report the overall adverse events, but just these immune-mediated hepatic adverse events, which is kind of a little bit biased. And that's all.
So
Any
what were
questions?
the symptoms that led to steroid induction? What was
The-
elevated ALP and AST? Stephen?
Yeah, exactly.
Can you show the Kaplan-Meier of the three treatment arms? What
Yep.
are the differences?
They don't show the differences. And
We don't know.
No, we don't know. I guess that this is a secondary endpoint, and this is not what they are trying to show here. This is my guess, because no, they don't show the log rank here.
So I chose to focus in only one abstract because this abstract is directly related to my actual project. So this is hepatic stellate cells promote regeneration after partial hepatectomy and toxic liver injury. So the first author is Tran Yin, and the last author is Robert Schwabi, and they are from Columbia University, in just close by. So this abstract was presented as an oral abstract in the Presidential Plenary Basic on during the Monday. So many data have provided clues about the mechanism and explaining the liver regeneration, especially after hepatectomy. So we know that very early after the 2/3 partial hepatectomy, hepatocytes receive a lot of signal, both mitotic signals like HGF, EGF, and also non-mitotic cytokine, which will make the hepatocyte enter in the proliferation states. And this process implicates the crosstalk between the different cell types in the liver. The first cell types which will react after partial hepatectomy are endothelial cells. So they will produce some cytokine, directing, we will target the hepatocyte, and inversely, hepatocyte will also communicate to endothelial cells. Also, stellate cells are implicated, and they secrete the very important mitogenic cytokine HGF, hepatocyte growth factor. And we also know that macrophages are involved in the process of liver regeneration. So this is a very complicated crosstalk between the different cell types that leads to liver regeneration. But hepatic stellate cells, whereas they are known as the key cells in fibrogenesis, the properties of activated hepatic stellate cells have been largely characterized, but the properties of quiescent hepatic stellate cells are less characterized, and especially the exact contribution of hepatic stellate cells in the context of liver regeneration remains poorly understood. So the aim of the present study was to assess the role of hepatic stellate cell in liver regeneration. So how did they do? So first, they performed the analysis of primary mouse hepatic stellate cell transcriptome using the Affymetrix microarray. So to do so, they isolate the primary hepatic stellate cell after partial hepatectomy. So they isolate them using the conventional method with the nicodens gradient after in situ enzymatic digestion. But they follow this step by a FACS sorting. So they use the specific property of hepatic stellate cells that they contain vitamin A. So they will be autofluorescent, and this autofluorescence will be visible in the violet FACS channel. So here you have the illustration of this FACS sorting. So this has been already published as a method that can improve the purity of the hepatic stellate cells without decreasing the different marker of hepatic stellate cells.
Is that after the gradient? Or it's
After the gradient. So you do the gradient, and after you FACS to increase the. This is also the method that we use now in the lab here. So this lab, Robert Schwabi lab, has already shown that LRAT is a very specific marker of hepatic stellate cells. Here you see the normal liver of LRAT-CRE-ZS screen mice. So all the cells expressing LRAT are in green. And here the red is staining for desmin, and you can see a perfect overlay. So LRAT is a good marker of hepatic stellate cells. So to deplete hepatic stellate cells in mice, they inject diphtheria toxin in transgenic mice, LRAT-CRE positive, and that mice have also CRE-inducible diphtherin toxin receptor, IDTR, and they are also CRE reporter tomato mice. So in these mice, all the cell expressing LRAT, this is the hepatic stellate cell, should also have a receptor to the diphtherin toxin, so they will be susceptible to react to the diphthery toxin, and they will be also stained in red. And as a control, they use the mice LRAT-3, but IDTR negative, tomato positive, and they inject the same toxin in that mice. So this is the design of their experiment. They have these transgenic mice. They realize the depletion of hepatic stellate cell by injecting the diphtheria toxin, low dose. And this will lead to kill only the cells which express the receptor, so the LRAT positive cells. And 7 days after, they induce the liver regeneration using different methods. I will explain this method in the next slide. And 48 hours after inducing liver regeneration, they will sacrifice the mice to study how the liver did regenerate. So the main 2 endpoints to assess liver regeneration they use were the ratio liver weight over body weight and the proportion of hepatocytes which were positive for the proliferative marker Ki67. So to induce liver regeneration, they did 4 different methods. They did the partial hepatectomy, 2/3. They did 1 injection of CCL4, single dose CCL4. They also inject acetaminophen, and they also inject TCP-OBOP. I don't know exactly the way you are used to hear that. This is a constitutive androstrain receptor agonist that induces the robust hepatocyte proliferation without any liver injury, without any hepatectomy. So also they did another step. They wanted to assess if the inhibition of hepatic stellate cell activation has an impact on the capacity to enhance liver regeneration. So to inhibit HSC activation, they delete the key genes of HSC activation in the LRAT-3 mice. That means they cross the LRAT-3 mice with either beta-PDGFR flox mice or TGF-beta-R1 flox mice, collagen-1 flox mice, or YAP flox mice. So 4 different methods to inhibit HSC activation in vivo. So here are the results. So this first picture shows you the efficacy of the model of depletion. So this is the control mice. The mice which have not which doesn't have the IDTR, but which receive diphtheria toxin. So you see all the red cells are the cells expressing LRAT, so the hepatic stellate cells. And in the experiment mice, we have which have the IDTR and which receive the diphtheria toxin. You see that the amount of red cells has dramatically decreased. So they said that 80% of hepatic stellate cells were depleted in this model. And these results were confirmed by a decreased expression of HSC marker assessed by a PCR. The mRNA of LRAT, N2, LHX2 were also decreased. And also, Robert Schwaber had this precious information during the basic seminar that this model of depletion wasn't associated with any morbidity because the body weight of the mice wasn't changed. He says, it is his words, in despite of extrahepatic expression of LRAT. So now let's look at the result about of this depletion of hepatic stellate cell on liver regeneration capacity. So here this is 2 days after partial hepatectomy. You have the ratio liver over body weight, liver weight over body weight, and you see that in the mice depleted for hepatic stellate cells, the red bar, this ratio is significantly decreased, 2.1 versus 2.8. So this, I found this illustration on the website from the AASLD, of the AASLD website. So I put all the same graph for all the other slides just to have an illustration. And also after partial hepatectomy, the mice depleted for hepatic stellate cells show a dramatically decreased in the proportion of proliferative hepatocytes, as assessed by the proportion of Ki67 positive cells. So the same result was observed after CCL4 single dose. The same result after TCB or BPOP. I don't have the figure of the result here. And the same result after acetaminophen. So these results were only shown in the written abstract, but were not shown on the oral presentation for the acetaminophen. So in the 4 different models of liver regeneration, either after surgery, either after toxic, or either after the TCOBP, without any surgery, any toxic, the absence of hepatic stellate cell lead to inhibit liver regeneration significantly. So next, they wanted to understand the mechanism. So they did the analysis of the transcriptome of hepatic stellate cell, the primary, and they show that there were no activation of hepatic stellate cell at 3 hours and 48 hours after partial hepatectomy. But at 3 days, 72 hours after partial hepatectomy, they show a very moderate activation because you see the marker of activation as a moderate increase, 3-fold, 1.8-fold, 1.9-fold. But conversely, the marker of activation of proliferation, sorry, the proliferation marker were increased in hepatic stellate cells. So this result enables them to conclude that after partial hepatectomy, the hepatic stellate cells are not really activated, but they proliferated a lot. So they wanted to see if they inhibit hepatic stellate cell activation, will it impact the liver regeneration? So they did the same experiment of the partial hepatectomy on mice, LRAT3, which are depleted for these specific genes only in hepatic stellate cells. And for the 4 different methods, they observe no impact on liver regeneration. That confirms the data of the microarray. That means that even if you block the activation of hepatic stellate cell, they will still promote liver regeneration because anyway, they don't activate after partial hepatectomy. And they also inhibit the deposition of extracellular matrix by performing a KO of collagen 1 in the whole liver. And the KO of collagen 1 did not impact also liver regeneration. So no need to have activated hepatic stellate cells, no need to have collagen to promote liver regeneration after partial hepatectomy. So this experiment was done only after partial hepatectomy.
This is all at 48 hours.
Yes, that's what they reported as the only endpoint. So maybe they try different endpoint as we discussed after the presentation because you asked them about the mortality, but they show only the time point at 2 days after partial hepatectomy. And the same, 2 days after CCL4, 2 days after acetaminophen.
Just that TCP or AOP.
Yes.
So what is it exactly? Is it promoting proliferation of hepatocytes?
Yes, exact.
So it's not really a regeneration connection.
It's a chemical. It's a chemical which induce hepatomegaly, but without any cancer transformation, without any injury.
Why do you tie that to regeneration? It's just a proliferation.
Because when you give this chemical product, the hepatocytes are getting into proliferation. So it's like another model of liver regeneration. Yes, it's another model of liver regeneration. And finally, they did we know that the main source of hepatic growth factor is hepatic stellate cells. And in their mice depleted for hepatic stellate cells, they saw that the expression of the mRNA of HGF was significantly decreased, as expected. So they did the partial hepatectomy in mice in which HFG has been KO only in HSC. Yes, excuse me. FGF has been deleted only in HSC because in the rat positive cells. And this leads to have no impact on liver regeneration. So even if you prevent hepatic stellate cell to produce HFG, you won't prevent them to promote liver regeneration. So that means that there are other ways explaining the mechanism implicated.
Did it quantify HDF levels in the blood or anything?
I think they no, because you think that maybe another cell type is taking the lead on the production. Yes, no, they didn't check that. But I know that you have also Yes, you have also endothelial cell producing HGF for sure. HFG. Oh yes, it's because it's a typo. Sorry, I was looking at the mice. So in conclusion, hepatic stellate cells are crucial for liver regeneration following partial hepatectomy or toxic injury. And as I say, I repeat, because I think the strength of this work, they confirm that in 4 different in vivo models of liver regeneration. This crucial role of hepatic stellate cell to promote liver regeneration is independent of their activation, independent of their ability to produce ECM, and independent of their production of hepatic growth factor. And they are doing other study right now to determine the pathway through which hepatic stellate cell promote liver regeneration, and they are specifically interested in Wnt pathway. Do you have some question?
Yes. So there is a very strong proliferation signal when you look at the gene, 60-fold increase.
Yes.
So there must be a very strong mitogen in place to induce that level of proliferation, right? And despite HGF is not involved, did they talk about any other potential mitogens that are involved?
You mean mitogens towards stellate cells?
Yes. Yeah.
No.
Did they talk about any other known, mitogen?
No. No, they didn't talk about that.
Yeah, that's interesting.
So a question and a couple of comments. Was the time course of proliferation gene induction the same as the time course?
They did not precise that point. I should have asked the question. Yes.
It's very interesting because, we've always...
yes, we don't know.
Well, we've always, conceived of stellate cell activation as a sort of monolithic response, and I was certainly naive, and it may well be that they respond differently in different situations. So
yes,
regeneration might have more of a growth-promoting signature in the cells, and injury might be more of a fibrogenic signature. And I guess it'd be interesting to do both, like CCL4 and partial hepatectomy, and see if there's a hybrid phenotype. But the other implication we're thinking about is if we get very good at either depleting stellate cells or antagonizing them in diseases where there's both regeneration and fibrosis, we may inadvertently block regeneration. It reminds me of the extreme example when sunitinib, which is a VEGF receptor block, which is very effective, was used in liver cancer, and a handful of patients died of liver failure. So if we're blocking proliferative signals, which might include blocking stellate cell activation, we might need to be careful that we don't induce failure to regenerate, which these livers are dependent on.
yes, for sure. For sure. Yes, they don't precise the timeline exactly. So maybe they choose the time point when the fold was the maximum, but I don't know when.
Yeah.
I don't know when. And you're right. Usually the activation and the proliferation capacities, they go together, but maybe it's more complicated in that we think.
Traditionally, the mitogens for stellate cells are PDGF, and for fibrogenesis is largely TGF-beta. So there are no different pathways, and I guess one can look at what the levels or expression of those two ligands are. They might be different, for example, so that you have more PDGF, not as much TGF-beta.
Yes, thank you for your comments.
Which actually sort of makes sense because TGF-beta is a growth suppressor, and the last thing you want to do if you're trying to regenerate a liver is provide more TGF-beta.
What was the number of that?
24, if I remember well. Let me check.
I think
Yes, 24.
Okay, so just to follow up to what Sarah was talking about, I just want to describe, first of all, 2 talks. The first one is just an abstract that talk about the, kind of following on to what Sarah was saying, that the Wnt might have a role in regeneration, specifically the Wnt that are being secreted by stellate cells. So in this abstract came out of the Paul Munga lab, they found that elimination of Wnt secretion from stellate cells was able to impair cyclin D1 at baseline, and also hepatocyte proliferation after partial hepatectomy. So again, it was presented by Paul Munga. They basically crossed this Wnt-less flox mice, which basically is the cargo receptor required for all Wnt secretion, effectively blocking the secretion of all Wnt from stellate cells, with LRAC3 mice, which they are described to be specifically expressed in the stellate cells in the liver. So this led to the complete blockage of Wnt being secreted from stellate cells, and they found that in these knockout mice there was reduced expression of several cyclins that are involved in proliferation, cyclin B1, B1, A2, following partial interestingly, they saw there were less BRDO positive cells at this 24 to 48 hours window following partial appendectomy, but everything rebounded by 72 hours following surgery. So suggesting that the effects of the Wnt coming from the stellate cells might be a temporary effect, or it could be compensated by other cells maybe stepping up to the plate and secreting Wnt in absence of Wnt coming from the stellate cells. And I think they mentioned in the talk, but didn't put in our abstracts, that overall there doesn't seem to be a regenerative effect in these mice. So that was really interesting. They didn't say exactly why, but I suspect that there might be no difference in liver body weight ratio, which is kind of like the global measure of how well the liver has regenerated. So this begs the question of whether or not it's actually the Wnt that's being secreted by stellate cells that's kind of contributing to the stellate cells pro-proliferative effect, or maybe it's something else that can be discovered. Yeah, so their conclusion was that other cells can compensate and increase secretion of pro-regenerative Wnt in this model.
Sammy, did they use an inducible knockout or constitutive expression?
Constituted, because they only have the RAC3, which is constituted, yeah. So any more questions? This is my first abstract.
No, just a comment. There's a company, this is probably known, that I work with called Conserved that has Wnt ligands, therapeutic Wnt ligands. And I think those are going to turn out to be very attractive as regenerative therapy. And the challenge when you provide Wnt is how do you prevent this from also driving cancer? So they're thinking about different clinical settings in which you could really use short-term regenerative signaling without long-term therapy that might induce liver cancer.
Yeah, another thing that Paul Manga mentioned was that there's something like 19 Wnt's or something, so there's a lot of different Wnt ligands that could be tested. But here I think it's nice that they kind of got rid of all of them and didn't seem to have an effect.
There must be some reason why we've evolved 19 different Wnt ligands. I don't know what it is yet.
Well, they're not all working.
Yeah, clearly there has to be cell specificity, contact specificity, but of all the molecules, I mean, 19 different ligands, it's really...
Yeah, it's interesting. I think it's going to keep Manga busy for a while.
Yeah.
They didn't show whether or not there was a presence of Wnt signaling in the livers, right? I don't remember them actually
What do you mean?
to read out whether or not there had been compensation by other cell types, which just occurs to me. It doesn't conflict with the idea that Wnt really produced by stellate cells could be active, required in regeneration. But if they've knocked it out starting from early development, it's clear that maybe other cell types have compensated by the time they reach it.
Yeah, so we don't know when the compensation could have occurred. Yeah, that's true. So maybe in a short-term model like we have with the depletion then of the stellate cells, then maybe there won't be as much compensation. So this is the second and last abstract that I want to present. So this is a talk from Frank Hackey lab in Germany. So he titled the talk Hepatic Macrophages in the Liver Regeneration, but he didn't mention anything about regeneration, basically focused on liver fibrosis. So basically hepatic macrophages in hepatic fibrosis. So this is just a summary of his talk. A lot of the work has been published already. So that's what I found at the AASLD. These big talks that were given, a lot of the stuff are not really preliminary. So he summarized his recent findings in this little diagram here. Basically, they found that there are Ly6C high monocytes that are derived from the bone marrow in the blood that can come into the liver in the presence of some kind of injury, and they differentiate into these fibrogenic, pro-fibrogenic Ly6C positive macrophages. So these macrophages secrete a bunch of inflammatory cytokines that contribute to stellate cell activation. So this is why this is considered a pro-fibrogenic macrophage. And what they also found is, interestingly, these macrophages, after a while, can also become these restorative Ly6C low macrophages. And these macrophages In contrast to the Ly6C high macrophages are actually restorative. So they are secreting ligands that promote the apoptosis and also the inactivation of stellate cells. So if your stellate cells activate, and then in the presence of these new ligands, these stellate cells inactivate or they apoptose. So the reason I'm interested in this work is because there's a ongoing interest now that looking at how stellate cells not only activate but also deactivate in the presence of fibrosis regression. So this is another photo that he shows. So he's talked a little bit all over the place, but all of them very interesting. So this is one more thing that his lab was doing. So they're using 2-photon intravital imaging to track these macrophages that are coming into the liver in the presence of injury. So this is actually a slide from Scott. I didn't take a picture, but he showed the exact same picture that Scott's taken a picture previously. So these, what they found is that here in the blue are natural killer T cells, and then all the red dots are macrophages that actively took up this fluorescently labeled latex particle. And these little lines here are actually tracking the movement of these cells. So this is imaging in the live mouse following injury of acetaminophen overdose. So you can see that the blue here is collagen, imaged by second harmonic generation from the collagen. And what happened is that these NKT cells are actively migrating into these regions that are actively being injured and are having these collagen kind of forming around these regions. And also there's also migration of the macrophages actively into these injured regions. So this is, I guess, better shown here. So the macrophages are also moving to these regions that are collagen rich. So this is kind of interesting because our lab is thinking more and more about this kind of possible communication between stellate cells, which are probably producing the collagen, and the macrophages, and the kind of communications they could have that kind of promote fibrogenesis. So this is an image that I took previously in the lab, not using live imaging, but using tissue clearing. So here in pink are the stellate cells, which we've mapped in 3D, and in the green are macrophages, which overlaid on top of the stellate cells. And what we found is that the stellate cells and macrophages are in very close contact. So see if this plays. Yeah, so the stellate cells again in the pink here, and then the macrophages are in extremely close physical proximity with the stellate cells.
True love.
So I thought that was a really novel observation, but unfortunately a paper just came out a few weeks ago that published the exact same observation, that the Kupffer cells are in direct contact with the stellate cells, and that they provided 3D imaging and also EM reconstruction to show that the stellate cells and the Kupffer cells are directly interacting. And their story is that the stellate cells, this physical interaction is required for the monocytes to come into the liver, and then after this physical interaction with stellate cells, this helps the monocyte differentiation into the macrophages. So this is what Taffy was kind of presenting in his talk. So we hope to focus on the other aspect of interaction to see if this interaction with monocytes or macrophages could affect stellate cell function. So this is how which I will integrate kind of these observations from other labs and try to use that to guide my own studies to see if we can mine single-cell RNA-seq data that's now available for potential mediators of this physical interaction, and see if we can maybe carry out a small shRNA screen in a co-culture system, stellate cells and macrophages, to see if we can break that interaction and what kind of effect that would have on the stellate cells. And then ultimately in vivo to see if that has an effect on how stellate cells behave in injury, in fibrosis, in fibrosis regression, and maybe contributing to any activation. Yeah, so that's it. Oh, and I can take any questions.
Questions? So for the last 15 minutes, I will present two different studies. The first looking at correcting the microbiome in PSC patients, and then the second looking at conserved mechanisms of fibrosis across different organs. So the first study was presented at the Presidential Plenary for Translational Science, abstract number 26. And they're looking at dysbiosis in primary sclerosing cholangitis. So if you were at any of the microbiome sessions at AASLD, you will have observed that people are looking at the microbiome in just about every possible liver disease. But the case for the contribution of microbiota to PSC is particularly strong. It's been observed that patients with PSC have portal bacteremia, so there's evidence that bacteria are getting from the colon and invading to places that they shouldn't be. You can also see that there's positive culture if you look at the bile, and that there's enterotoxin building up in cholangiocytes. Furthermore, if you take microbiota from these patients and culture it and then expose naive lymphocytes, you get a Th17 helper immune response, which is very close to the kind of autoimmune profile observed in PSC patients. So this is an area where there's a pretty strong case to take a look at the microbiome and examine whether or not it has a driving role. And this is a publication on the left-hand side here from 2015 during a principal coordinate analysis, and it shows you that there's a unique dysbiosis in PSC. So these are patient samples. Their colonic microbiome is distinct from patients with inflammatory bowel disease and from healthy controls over here on this element. So the abstract that I'm presenting is really building on a really strong paper presented in Nature Microbiology earlier this year that identified Klebsiella pneumoniae as the pathogenic microbiota in PSC patients. And they very nicely characterize that if you take microbes from PSC patients and culture them, you'll get a number of different cultures that grow out. It's a complicated mix. If you put that into a germ-free mouse, those microbes will invade into the colonic epithelium. And that's not seen in a pathogen-free germ system. And through a process of elimination, they're able to show that Klebsiella is really the bacterium responsible for this invasive property. If you take it out of this mix from PSC, you're not going to get invasion. If you take a look at a monolayer culture of colonic epithelium that has had Klebsiella placed on top of it, you can see exactly how it's doing this. It's actually inducing apoptosis of that and creating these little pores, which are then providing the route for invasion of all the other microbes. And really importantly, they showed in a model of PSC that if you remove Klebsiella itself from the mix, but leave the rest, and here they've done that by taking the PSC mix and putting in a Klebsiella variant that's not capable of forming pores, that alone is able to prevent invasion. And then furthermore, in a model of PSC, it normalizes liver injury, looking here at ALP, and also reduces that IL-17 autoimmune response. So this really set up the idea that if we could somehow target Klebsiella in many of these patients with PSC, that you'd be getting at the major contribution from the microbiome. So the idea presented by this international group was, let's use bacteriophages to knock out Klebsiella specifically. And I've put up a picture here of the classic T4 bacteriophage. A really nice setup for the study. First, they wanted to characterize an international cohort of patients and really get a sense of how many of the patients with PSC harbored a pathologic KP. And you can see they had a really large cohort, 420 patients from Germany, 120 from France, and then another 112 from Israel. They characterized their stool samples, and then they had a library of KP that they grew up in plates. And the idea was kind of just expose those plates of grown KP to environmental samples and see if we can mine the environment to find bacteriophages that were capable of killing, which I thought was fairly elegant. And they didn't describe what their environmental samples were, so we'll have to wait for the paper to come out, but I don't know if they were just looking around their lab or in their front yard. The results are very nice. So they were able to show, really confirming the prior literature, that KP is enriched in the PSC patients that they sampled, and the presence of these pathologic strains are correlating with disease outcomes. They isolated from their environment more than 20 phages that were able to kill KP, And then they're mixing and matching to make up more than 100 different kinds of therapeutic cocktails that they could deliver. And they've screened both in mice and in vitro for the most potent killing capacity of the bacteriophage cocktails. In their abstract, they only presented the data at left, which is just looking in bacteria cultures on a shaker for killing capacity. But you can see that they had a range of killing efficiency from this kind of middle group here, where many of the phage cocktails are, to a really nicely optimized couple of cocktails here. So I think they've taken a really strong publication from earlier in the year, but developed what looks like a fairly robust strategy for moving this into the clinic. And this caught my eye in particular because there was a Nature paper out last week showing a similar approach for targeting pathologic bacteria in alcoholic liver disease. So I think this is an area that's pretty hot.
Why always the cocktail? Just to prevent resistance?
They commented on resistance and said that it was low, but they didn't justify the use of a cocktail rather than one bacteriophage. So that's a good point.
Just to remind everybody, bacteriophage are viruses that infect bacteria.
Right.
So would this have to be a lifelong therapy, or is it a one-shot thing and it changes the microbiome?
Yeah, so I think the question there would be, why did the PSC patients harbor this pathologic strain? Is it some environmental exposure, or are they particularly able to harbor that? And I don't know the answer to that question. I think it's a really pertinent question, and it depends whether you have some exposure to a pathogen one time and it takes root and you wipe it out with one therapy. If instead you are a uniquely susceptible incubator, then maybe you need repeated therapy.
The problem is this bacteriophage will also destroy the other bacteria. And in PSC,
They're bacteria specific.
but
Well,
they didn't test for that.
Yeah, they didn't describe in their abstract whether or not it was selective for KP. They optimized it for killing that pathogen, but yeah, it's a good point.
Because also there is data supporting that there is like good bacteria for PSC.
Yeah.
And especially in germ-free mice, if you induce a biliary disease, it gets worse. So maybe you need some good bacteria also.
Yeah, I think it's really, it's going to be complicated. It's not unlike the fact that lots of people harbor, methicillin-resistant strep, and they don't get sick, and so it's always, it's going to come back to the story of host and environment. Do patients, you said, do patients with PSC have an immune defect that makes them permissive for this bacterium, or does the bacterium take root and change the immune system? It's probably a little both, but there are certainly immune markers that are associated with PSC risk, or genetic markers, so there's clearly a genetic background. To answer your question, Charlie, it's not known yet how long this treatment would be, but there are isolated cases of what's called autobrewery syndrome. It sounds ridiculous, but it's actually real, where patients harbor an overgrowth of a bacterium that produces alcohol, and they literally, they've been arrested for drunk driving, even though they're not drinking. And so in patients like that, they've been treated with antibiotics and fecal transplants.
But that's more like an infection, right? It's not a, because like the microbiome, the bacterial microbiome is sort of thriving environment, whereas it's not an acute infection.
Well, the autobrewery syndrome, it's very much like this. They're able to harbor certain strains of bacteria that convert sugar into ethanol, and those persist over time. So it's not like an acute infection. In fact, it was just...
Right,
yeah.
They don't have the bacteria in the blood, so they are not infected, they're just around the...
Yeah.
All right.
We should probably move on, yeah. All right, so the second abstract was actually a poster, and it's looking at a particular type of fibroblast that's responsible for organ fibrosis. This comes out of David Brenner's lab. So by way of background, portofibroblasts are a minority population of non-parenchymal cell within the liver. They make up only about 0.1% of all liver cells, but they have really important functions in supporting the structure of the biliary tree, and they are major contributors to fibrosis, especially with a certain type of biliary injury. And David Brenner's group has published previously that there are particular markers associated with activated portofibroblasts, particularly mesothelin and Thy1. So the question that they had in advancing this study was, are there kind of shared programs of portofibroblasts activity outside of the liver. It's really more of a conserved program in many organs that are susceptible to fibrosis. So I'm showing you here data that was on their poster, but it was really well characterized in the JCI paper in 2017, working out a regulatory network where mesothelin and Thy1 are participating to regulate activation of portal fibroblasts. So the way it works, you have TGF-beta R1, a really important receptor responding to TGF-beta in the activation of portal fibroblasts, and that is constitutively inhibited to a certain extent by Thy1. Mesothelin, however, is able to come in and inhibit Thy1 and de-repress the TGF-beta receptor, allowing it to respond more readily there. So as you can predict based on that network, if you knock out mesothelin, you're going to get stronger repression of TGF-beta receptor, and that will tamp down fibrosis. The opposite is true, though. If you knock out Thy1, you would expect to have more robust fibrosis, and that's what they were able to show here in their MDR2 model. Just over here, you can see without mesothelin, really attenuated fibrosis, opposite story if you knock out Thy1. So they've exported this system and said, can we injure other organs and play with these two regulators in the same way, and would it produce the same effect? So looking first at kidney, where they did a unilateral ureter obstruction model, looking at mesothelin knockout. Green is collagen, red is Thy1, and I think it's pretty apparent that there's a lot more collagen deposition here in the wild type versus less when you've knocked out mesothelin. Again, opposite story is true here, looking at serous red staining for collagen deposition. If you've knocked out Thy1, there's quite a bit more robust staining. And then showing that these are acting along the same pathway. If you do a double knockout for mesothelin and Thy1, you kind of rescue back towards the wild type. They also extended that observation to the lung using a bleomycin model. There's some mislabeling on this poster. So I'm not 100% sure that all of their data isn't somehow carried over. This does look like lung to me. This was actually labeled as a ureter model. But you see here on the left-hand side, Thy1 knockout certainly increases fibrosis. And a double knockout appears to rescue. I'm not as convinced by their mesothelin knockout here, where the serous red staining seems to be, I guess, slightly attenuated. She looks a little bit worse than the mesothelin knockout. So they also did RNA-seq to compare portal fibroblasts to the fibroblasts that were Thy1 positive, isolated from these two organs. The photo that I took of it was so poor that I didn't want to put it up, but they concluded that there were kind of similar gene expression patterns between these, supporting the idea that there is a common fibroblast population seen across these three different organs.
So that's not the only pathway. Because if the double knockout, someone's in Thy1.
Certainly it's not the only pathway, but it is important, I think, in activating.
Well, I mean, with the mesothelin, it's doing something else besides Thy1. Because if you're missing Thy1, right, that's Thy1, that's a double knockout on the right.
Right.
Yeah, so it shouldn't matter if you.
It should be like the wild type. You know what?
Yeah.
I
I think we are running over time. Yeah. Okay.
Okay, so thank you guys for the effort. It was
Great job.
Thank you.